So our final presenter today is Dr. Mandy Bish. Welcome to Illinois.
Thank you. Thanks.
She's an extension specialist at University of Missouri, MU. She currently coordinates MU's Integrated Pest Management IPM program, serves as the lead for MU's Extension Field Crop Pathology team, and directs MU's SCN Diagnostics. Diagnostics, which analyzes approximately 2,800 samples annually. Mandy is a Missouri native, and she grew up on a farm operation in mid-Missouri and earned her BS in plant sciences at MU. She then earned her Ph.D. in cell biology and molecular genetics at the University of Maryland in 2009. And Mandy worked as a research molecular biologist at the USDA-ARS Foreign Disease and Weed Science Research Unit at Fort Detrick, Maryland, before returning back to her Missouri roots. She has worked at the University of Missouri since 2014, where she has studied row crops and pests. So please join me in welcoming to Illinois, Mandy.
Thank you.
Good morning, everyone. Like Stephanie said, my name is Mandy, and I am from Missouri. Y'all have the better soil, but we have the better basketball team this year. So I just want to put that in. I don't get a chance to say that very much, okay? You know, I have to hear I-L-L-I and I all the time, but M-I-Z-Z-O-U today. No, I digress. So I do want to point out that I took over a bunch of roles in April at the University of Missouri following some departures of faculty, okay? So I've been the director of SCN Diagnostics since April. I am going to present a fair amount of research, but I would encourage you, if you have questions during it, please just ask me during, you know, it doesn't do us any good if I don't explain it well. So please don't hesitate to just interrupt me and ask questions. I do like to show this slide if it'll play, because how often do you get to see SCN under the microscope? So these are eggs hatching into the juveniles, and it's that juvenile stage that's going to go through the soil and find a soybean root to infect. So just a different view of SCN from the lab. And up front, I want to thank a bunch of partners who have been invested in researching SCN for a while now. So Jeff Verizone, if you have ever sent samples to our clinic, he is the senior research specialist there that you probably interacted with. Caitlin Bissonette was at Mizzou before taking a new position and worked closely with the SCN Coalition, the United Soybean Board Partnership. And then John Lloyd runs the Missouri Strip Trial Program, and I'm going to share some results from that. And then we just have a lot of undergrad and students invested in this. And I also want to say, Mark was pretty modest when he said that he helps undergrad students. If you have high school students that want to go into agronomy, Mark has an excellent track record, okay? Don't tell my administrators I said that, but Mark is excellent. And so I try to steal our undergrads for this research. And this would not be possible without Missouri Soybean Merchandising Council. They invest a lot of money in this initiative. And so first, just that brief background and overview, that big picture. So SCN, you know, it costs a substantial amount of money annually. And oftentimes this is, Jim said something kind of like this earlier, but about SCN specifically, it's usually small increments of yield robbage. So we don't necessarily notice it's happening. So it could be, I got 60 bushel per acre beans, and you could have gotten 65 bushel per acre beans potentially if you had a problem that you weren't aware of managing. It was first detected in North Carolina in 1954. The second state it was identified in was Missouri. So y'all got water hemp, we got SCN. I guess we all will share. The eggs can persist in the soil for years. Does anybody have a guess at the longest recorded time that an SCN egg has been in the soil and still hatched or been viable?
7.
7? 7? Good guess. I would have gone somewhere similar. It's 40 years. So once the pest is present, it's not necessarily going to go anywhere. It's going to be there to stay. So, you know, initially back in the 50s when this first started showing up, the idea was that you had to have symptoms on the leaves that you could see for it to be a problem. Obviously now we know that that is not true. You can have yield losses even when symptoms aren't present above the ground. And I'm going to share, you know, most of the data that we have on SCN is taken by two different things. So one is the egg counts. So we do egg counts for many of our assays to determine densities, populations of SCN, and how different treatments work. And this is just an image of one of those eggs and a juvenile inside it. The other thing that I'm going to share some about is our females. So SCN females form these lemon-shaped structures on the roots, and we do a lot of female counts on the roots. Okay, so when I'm referring to the female counts, now you can have a little bit of an idea of what that is, and then the egg counts under the microscope. Okay, so when I first came into the position, I had some nice data that was generated previously, where they just did a lot of small plot research, and we're looking at SCN egg counts and end of season yield. So pretty straightforward. Okay, so on this graph, the SCN egg counts are on the x-axis, and the yield is on the y-axis. And this is probably 6 trials in 3 different regions of the state of Missouri, and it's about 250 different data points. Okay, and so that's what this trend line is showing, those are the trend of those data points. Now, some of this looks pretty substantial. I will say that most of the samples that we get in our clinic right now, they're not at 0, but they're within the probably 2,000 to 10,000 eggs range. Okay, so you know, how many of you have dealt with glyphosate-resistant weeds? Anybody in here? Yeah. So if I'm kind of using an equivalent, I wouldn't say, you know, we're not at the point where it's like we've lost 96, you know, we're having to spray 96 fluid ounces to try to control something, but we're starting to see like an increase in the number of eggs, meaning that our management is starting to decrease. What we don't want to get to is we don't want to get down here, because the management options for SCN are far and few between. And I'm going to go through some of our research that kind of summarizes some of the management options that we currently have and some of the things coming down the pipeline. So SCN from 1954, you know, this map just shows where it is in the US, where it's been documented. Illinois has been identified in every county in the state. Missouri has this little nice white patch here indicating that there's no SCN, but that's the Ozarks. So there's not really, yeah. Otherwise, so it's found in every soybean growing region or county in Missouri also. So a little bit of the background. The first management was relying on genetics, which is a good place to start, right? And so there was a lot of work in identifying soybean genetics with resistance to SCN. And there were a few identified successfully. And the most commonly commercially available ones are called PI 88788 and Peking. Okay, so these are what we refer to as the sources of SCN resistance. One of the problems we have is that greater than 95% of commercially available soybean varieties rely on PI 88788. Okay, and we've relied on that for over 3 decades. So we used to be at the very low populations of SCN in the fields, but what's going to happen anytime you expose the pest to the same control method, right? And that's where we are starting to see this breakage. One of the things that we do in the lab is we say, okay, well, how well can this SCN population, how well can it reproduce on these varieties that have PI 88788? You know, and that's a measure of whether they're developing resistance or not. So in Missouri, I don't know how many fields were sampled in Illinois, but it's 88%. So I'm going to walk you through this. In Missouri, in 2015, we went out and sampled about 250 different fields. Okay, and about 70% of those fields, we found SCN. So it was just SCN present or not present in those fields. Then from the fields where you had SCN present, we took those populations and we measured 40 of them as to whether they could reproduce on the PI 88788 variety. 100% of those 40 populations could reproduce on PI 88788. And so the number is 88%. Now I'm not sure how exactly the Illinois method was done, but it would be something similar. So we have a high reproduction of SCN on these commercially available lines. And so again, I just want to emphasize that I don't want to like, we're not here yet. We're closer, sorry for the people online, we're not, you know, down to the 40,000, 45,000 eggs consistently. We will see a couple samples that come in that have that many, but most of our samples are really in that 2,000 to 5,000 right now. And so how can we push that back closer to 0 is the question. You're not going to eradicate it now that the pathogen is present. But again, we do not want to move further down the x-axis, increased eggs. And so this is a slide by Dr. Greg Tilka in Iowa, just to demonstrate how quickly SCN can expand within a field. So if you had 100 SCN in your field, let's assume half of those are females, and then they have about 250 eggs. Each female has 250 eggs. Okay, and then the next generation, you take half of those, multiply by 250 eggs. And so the numbers in that second column show you the increase in SCN that you can get rather rapidly. Well, that's with 100%. That's if every single SCN egg is viable. Now, if there's one good thing about this, is that not 100% of eggs are going to be viable and germinate and hatch and find a soybean plant. So let's say 97% of them are not viable. Okay, so only 3% are viable. Still, by the second generation, you have 46,000 eggs. Now, if you go back to my previous slide, that equates to, at least in our small plot data, about a 30% yield loss, which is what Iowa continually says that they are seeing consistently in some of their fields, is a 30% yield loss. And these are just numbers from Greg's lab. We've never seen a number this high, 334,800 eggs, but the fact that you could go so long that would be a number that you see is pretty impressive and not necessarily a good way. So I like to share that.
Is it one generation per growing season?
Oh, that's a great question. So the question is it one generation per growing season? And no, it's so they can complete their life cycle in about 35 days. So you can see, yeah. Now you do have to have the host present. Okay, so I want to talk a little bit about Peking, because we talked about the PI 88788 resistance and that 95% of our commercial varieties have that source. So what happened to Peking along the way? Well, we also know that where PI 88788 causes SCN to die off, so the female SCN is going to attach to the root. As it starts to form its syncytium, that's where the PI 88788 is going to affect its growth and it's going to die. Okay, so the female dies, it can't reproduce. Well, Peking might offer some more benefit because it works as the juvenile. So you have your egg, it hatches, and as the juvenile attaches. So the idea is that Peking can control both female and male SCN, whereas the PI 88788 that we use typically only controls the female. The big question about Peking and why, does anybody know why did we put Peking on the shelves?
Yield.
Yeah, the yield drag associated with it, the lack of yield, right? And so once you have something like that happen, it's hard to get over that mindset, right? Once you see a yield drag once, it's over. Well, that was in the 1960s and 70s when they were developing that. And so Dr. Tilka has been pushing seed companies to get Peking into commercial varieties. And so this is a slide from his group. The Peking, this is a yield slide. Peking are in green on the very left side. All of his soybean lines that have PI 88788 are in the gray. And then the susceptible lines are in this gold over here. And there's a couple things here. Number one, these are heavily infested SCN plots. So you can see in the box, the PI 88788 had 14,000 eggs on average per strip. And the susceptible, the PI 88788 had more eggs than the susceptible had on average in each strip. While the Peking had reduced eggs, and you see the corresponding yield. So the two Peking varieties, about 70 to 72 bushel per acre. Whereas you can see all of these PI 88788 average, or there's a 50 bushel per acre, so somewhere between 60 and 50 bushel per acre yields again. And so these are in the same field, right? These are all taken in different regions of Iowa, but all of these varieties are being grown in the same field. And so again, it might be a matter of, well, we kind of expect a 60 bushel per acre yield out of this field, but you might actually be able to get more yield out of it. Knowing your SCN population would be helpful. So this year I have gotten some Peking seed to do some studies on PI 878 and Peking rotation. It's really hard to get Peking seed. So I got one bag in a maturity group of 3.1. Now in mid-Missouri, we're looking at 3.8, 3.9, maybe even a 4. But I took what I could get. And I'm just saying that to you all because if you do have SCN in your field, and if there is a need for another type of genetics, that push has to come from you all to get the breeders to work on it. And they are expanding. So 3.1 is better than last year's options, which I think ended at 2.8. So they are getting there, but it could use a little bit of push if you do indeed find out that you have an SCN problems. And so a lot of this again goes back to PI-88788 was working and there were no above ground symptoms. So there was not this demand to have additional genetics. So just take home points from the breeding side. Is Peking resistance available in the right maturity group? You know, and is the yield comparable? Most of the recent studies say the yield is comparable. All right, let's go to the nematode protectant seed treatments. And this was work, and actually Illinois had some data in this work. It's a multi-state study that was done over the course of maybe 3 summers, maybe 2 summers, funded by North Central Research, Soybean Research Program. So if PI-88788 works on the female and Peking resistance works as the juvenile is attaching to the plant, these seed treatments work at various stages. And the thought is that they may work as the juvenile is hatching, and some of them may work as the juvenile is moving to find the soybean plant, but it offers another place where the nematode can be controlled. And so this group, and this was done by my predecessor, Caitlin Bissonette, but they took different seed treatments and they treated a PI-88788 soybean. So a soybean that was resistant to SCN. They treated them. They introduced a nematode population that was resistant, though, to PI-88788. So you have resistant soybean, but you also have SCN population that's resistant to it. And they found some consistency. So it's not my job to push products or anything, but ILEVO was very consistent in all these trials, and most of these had some effects. So they were looking at SCN females, okay, and then you have your seed treatments along the x-axis. And these are your controls to compare to. So you have a PI-88788 without any seed treatment. And the R's, the B's are the letters. And most of these treatments showed a reduction in the number of females relative to that PI-88788. And then here's your susceptible there. Okay, so this was good. This is a greenhouse study, though. So what happens when we take it to the field? And in the field, the seed protected treatments were not near as consistent. This is just 3 fields from different states. And basically the non-treated is always on the right. And this again is the female count. So you can see in many instances, the non-treated had less SCN females than the treatments had. So there is promise in these treatments because they look so good in the controlled environment, but we need to take it to the next step where we can figure out what that environment is or how to keep the molecule around longer so that it has those effects.
Mandy,
Yeah.
you think it has to do with the root zone?
So the question is if it has to do with the root zone, and that is a good idea. So you're protecting the seed, right? And then the females are going to be 25 to 30 days later. So a lot has to go right actually for the nematode egg to run into the seed treatment, potentially because of the root zone. So you have these seed treatments. Again, this just summarized, there were 51 different environments where this was looked at and less consistent in suppressing SCN. But what we like is that there seems to be a yield increase in some of these treatments above the non-treated seed. So even though we're not suppressing SCN, we're seeing a little bit of a yield bump, right? Which isn't a bad thing. We like yield bumps, but I'm afraid we might be seeing a yield bump and assuming our nematodes are being managed when they're not. And so we did again, prior to my time, Missouri Soybean Merchandising Council sponsored an on-farm strip trial where they went out to farmer fields and did strips of ILEVO treated seeds and just a base treatment for comparison. Okay, so it provided some good data. Unfortunately, it's only ILEVO though. It's really hard to get people to start planting different seed treatment. It takes a little more time for planting and harvesting, but they did it throughout the state. And this graph was put together by Dr. Lori. It's kind of hard to see the green. The green diamonds represent the mean of each field. So each row is a location. The green diamonds represent the mean of the ILEVO treated strips, the yield of the ILEVO treated strips compared to the non-treated strips for each location. If you take, and then the yellow dots are individual yield for individual strips of ILEVO treated seed. And the black dots are individual strips for non-treated or base treated seed. If you take all of that together, this green dot at the bottom shows you that on average, there was about a 2 bushel per acre increase with ILEVO across the average field in Missouri under the average conditions, whatever those are, compared to the non-treated. And then if you had sudden death syndrome in your field, we had 3 locations with sudden death syndrome. You actually saw a larger difference of 3.5 bushel per acre in the ILEVO treated strips, which kind of makes sense because Fusarium is a soil-borne pathogen. It is linked to SCN to some extent, but you see an increase when you have pathogens present. So I don't necessarily want to paint a bad picture of any of these products, and some of them are helping with yield, but I still question their value as far as the nematode protectant and what are they doing to our nematode population. If you haven't seen any of that ILEVO data, this QR code will take you to our extension site that has the seed treatment trials. It also has the summary of some trials out of Iowa. So there were different results from Iowa. They didn't see quite the yield difference. They saw about a 0.6 bushel per acre difference in the strips. So the take-home points with the protectant seed treatments are, they can suppress nematode reproduction, SCN reproduction in controlled settings. They do it quite nicely and quite consistently, but it's not consistent in the field. So are we really, again, that concern of are we really managing our nematode populations with these seed treatments? Old reliable crop rotation. If there's a silver lining for SCN management is that corn is not a host. And I don't see I see that as something that the nematode is probably not going to develop resistance, be able to grow on corn. Famous last words. So just, when I took over in April, I was like, well, let's just go see what our current population is. We don't know. So thankfully the undergrads went out to different regions of the state and they took samples in the spring behind corn. Corn was grown in 2021 in some of the fields and soybean in 2021 in some of the fields. Then they went back in the fall after the opposite crop. So each of these fields, there was a crop rotation. It's just some of them had corn in 2021 and soybean in 2022 and vice versa for the other. In general, the counts across between spring and fall were pretty similar. So these are just bins on the bottom of egg counts. So you can see there's actually a decrease in eggs in that smaller bin of 0 to 500 eggs. This is an egg range that we're really not too worried about, though, to be honest. But if you look at some of these to the right, that 501 to 2000 and these, you do see an increase from the spring to the fall. So you had lower populations in the spring, higher populations in the fall, which would suggest that there is some level of SCN being able to reproduce on those plants. And when you break that down by the different corn and soybean rotations, I just like this because it just illustrates the point that corn is a non-host. So if corn was planted in 2021, then we went out and took spring samples at about 1,600 SCN eggs per 250 cc soil. Then we planted soybean in 2022. We went out in the fall and took eggs, and you can see that there is an increase. And all that soybean, I'm assuming, is PI 88788, just because we don't have the maturity groups for Peking those. So we do see that increase when we switched to soybean. Likewise, if you went from soybean to corn, there was a decrease across all the populations. So just not a lot of just averages there to show you that corn can still work in the problem areas. It's probably not as much of a problem as to Illinois, but we kind of get into that soybean on soybean on soybean on soybean rotation in Missouri. So
It happens in the South.
okay, all right. So it's a southern thing. Like, okay, all right. Cover crops and SCN. So I don't have any data on cover crops, but one of the things that the SCN Coalition was pushing when I first came in was annual ryegrass. And if I say annual ryegrass to a weed scientist, most of it, yeah, exactly. You're like, wait, Mandy. But Dr. Tilke's group up in Iowa did some greenhouse studies initially to look at SCN with different cover crops. And this is kind of where they got their annual ryegrass. And so they just planted, on one study, they just planted the cover crop into the pot and then looked at the female reproduction, SCN reproduction on those hosts. And so here's your soybean that is a host, and you can see there was a high level of SCN nematodes. And then for most of the cover crops tested, all of them actually had less than a 1 reproduction factor. They were substantially less than the soybean and similar to non-treated. So in a greenhouse setting, that was pretty good. If you can't see, there are mustards up there, there are mixtures, cereal radish, and annual ryegrass. So then that was kind of to see, what, trying to simulate what might happen over winter when you have that cover crop in. Well, then what happens in the spring? So they took these cover crops out and they winter killed them. Then they planted soybean into the same pots and measured SCN again. And here's your soybean on the right. And you can see that a lot of the SCN populations for a lot of those soybean following cover crops were similar or comparable to the soybean alone. But you see this reduction with annual ryegrass and cereal rye. Okay, so that's kind of where led down that path of annual ryegrass. But I can't promote annual ryegrass. So what we do have, because of Missouri strip trials, is we do have it says here, 218 samples from Missouri strip trials of cereal rye versus no-till throughout the state of Missouri. And if you just take those and look at the egg counts in those, you can see it's not statistically different. So I can't say that cereal rye reduces the number of eggs, but I can say at the minimum, cereal rye is not enhancing SCN. Does not seem to be serving as a host. We are trying to get our hands on some cover crust to do some work with that, to just kind of tease that out a little more. We're still trying to get some seed for that. Okay. So just rotation to corn is helpful. It's not a silver bullet. Cereal cover crops do not seem to enhance SCN reproduction at all. Okay, so what's coming down the pipeline, right? What is the next tool that we're going to be able to use? Just like any pest, reminder that new tools are not silver bullets. We have to steward them well. So transgenics, the ASF has been working on a transgenic crop. So the beauty of transgenic crops is that you can get the gene of interest into the plant faster than through traditional breeding routes. Okay, but the negative is that sometimes there's some regulatory bottlenecks that you can't be avoided. So they took their new transgene and they put it in a commercial control that has PI 88788. Okay, so you have PI 88788 with this transgene in it now. And they're looking at the SCN females again. So the left box in each of these is without the transgenic, and the right box includes the transgenic. So you can see there's a reduction in SCN in almost each of their trials that they did. There was a reduction in the number of SCN when this transgene was present. Okay, so that's promising. This is in the greenhouse. I'm not going to show the field data that they published from that study yet because I'm going to give them the benefit of the doubt that it'll get a little bit tighter than where it is right now. So this could be a potential tool that can be added to the toolbox, but it's not going to be available till the late 2020s. So 2027, 2029 is what I hear thrown around. Additionally, there's a lot of breeding efforts that go into this. And you all are losing a really good plant breeder too this year, I think, who's been very essential in some of this. But there have been a couple of new genes identified that our breeders are working with, but it can take just as long to get those new genes into the varieties that you want to grow. So I just want to wrap up with scouting for SCN and looking for an invisible pathogen. As I mentioned earlier, we typically do 2 types of assays. We're going to count SCN eggs. We're going to look at the females, and there's good and bad to both. And the females are something that you can look for yourself. You don't have to send in. I did include a sample submission form just to remind you that these services are fairly inexpensive. $25 for doing the egg counts. And then again, the females, looking for females is free, or however much you want to pay a scout to do that. So the egg counts, it's a little different than traditional soil sampling. You're using a probe that is 1 inch in diameter and 8 inches deep, okay? And you want to get close to the soybean roots if you're doing it in season. And if you're doing it, we recommend in the fall, because that's after, that should be the highest number. So when the female dies off, the eggs, a few of them are released initially, but most of them stay intact in the female's dead body, a cyst. I should have said that at the beginning, overwinter in that. Okay, so when you do a soil sample in the fall, you're going to get the cyst. We're going to crush up those cysts and count the eggs that come out of the cyst. Okay, but it's 1 inch by 8 inches soil cores. And then there's a couple different recommendations. If you've never had a history of the SCN problem, you don't know what your numbers are, then you just want to divide your field into grids of about 20 acres, take about 15 to 20 samples, walking in that W pattern, and combine them for a composite for every 20 acres. Okay. If you have an area of field that's low-lying or that is, you've seen SDS in it, that would be an area that you probably want to take a separate sample of. So take 15 to 20 samples from within that area, and then another area that doesn't seem to be problematic to get a comparison for what your levels are. Because if you have consistent SDS, I'm willing to bet that you have some SCN populations in there. One of the most frustrating things about SCN sampling is, any guesses how far these nematodes move in their lifespan? Maybe an inch. Okay, they're not moving very far. So the biggest complaint I get is that, you know, there's a high chance of missing the nematodes, right? So what I've started telling people, and this could be me being naive, I haven't been in the position since April, but is, you know, if you haven't seen it, that's good. That's good information to have as a baseline. But it's when you start seeing it consistently in your samples that we know that there's a problem, right? If it's that hard to sample and you start seeing it consistently in the samples you're bringing in, it's a pretty good indicator that there's a problem, just on the nature of how difficult it can be to get these. So if your soil probe goes in 2 inches, if your soil probe goes in one place, you might get 0. If your soil probe goes in 2 inches over, you might end up with 5 cysts. If each of the 5 cysts have about 200 eggs in it, then you have 1,000 and 0. So there are some limitations there. We're working to move towards molecular techniques that can be more thorough in detecting the SCN. But until now, I think a baseline information is good to have here. That I don't know if we really need that. I just was curious in our variability, like how much does a sampler affect the variability in responses of SCN? So my undergrads were looking for something to do. So they each went out to the same area of a field that we have cultured SCN in. So it is a field that we know has SCN because we do our research in this field. And they each took 20 cores, combined it into one sample, 20 cores, combined it into a second sample, 20 cores, and combined it into a third sample. So each of them had 3 samples, 60 cores total. And then the average SCN egg count is shown in the gold bars. And so the averages, I mean, if you look at that, 1,200 to 2,000 is going to put you in about the same bin as far as yield loss estimates go. So that wasn't too bad. But if you look at the error range, I have one student who might need a little bit more help with it. But there can be variability. It's just a point to show that there can be variability. I'm realistic. It can be a challenge. Yeah, it can be a challenge to sample for. I won't show this video, but this is just a reminder to tell you. So part of our IPM effort is to reach the next generation, right? So we have a TikTok account. We have some videos. If you're curious about the SCN lab or what it looks like, this video just goes through that process of grinding the eggs and I'm not going to share it because it's too loud, but you can find it on our YouTube channel. If you're not a TikToker like myself, I'm not. I hired an editor. Okay, all right. So take home points from that part. You know, soil sampling for SCN, it's relatively easy and inexpensive. It can be extremely frustrating, but inexpensive. And baseline information is going to be helpful for monitoring the extent of the problem. You know, if you've sampled and you haven't had any come back with eggs. So out of the 22 fields we did in the fall and the spring, we had 4 fields that didn't have any eggs come back in any of their samples. So that was pretty good. So I'm like, okay, well, maybe just wait like 3 more years before you spend money sampling again. But for some of those fields and farmers who didn't expect to have a problem, they had over 10,000 eggs come back in every sample. So I think a baseline information is helpful. And then the female counts. So these would have to be a little more timely. They're free, but they're a little more difficult. So about 5 weeks after planting is when that cyst is going to form. That female is going to form the lemon-shaped body. Sorry, it's not a cyst yet, but the lemon-shaped body that I showed on that first picture. You can't hardly see it there. If you're going to look for females on the plant, you need to dig below the roots. If you pull the roots up, they're going to fall off. Okay, they're just attached by a sucking device on the root. So you need to dig and cleanse. But yeah, about 5 to 6 weeks after soybean planting is when you want to look for that. And this is just a comparison to show you there's a nodule there. This is Dr. Tilke's sliding in. And then you have some of the SCN females for a size comparison there for what you're looking for. So finally, if you have Peking available, another service that is offered by most of these SCN clinics is what's called HG type testing. So we can test the nematodes in your field to determine whether they have resistance to PI88788, whether they have resistance to Peking, based on how well they can reproduce on it. So that's what this is, the HG type test is about. But what I've been telling our farmers, and I'm probably gonna get in trouble for it, is if we don't have another variety available, then it doesn't necessarily give you any helpful information, because what are you going to do? Go back with PI88788, right? So if you have Peking available and are interested in that, that's another service that we offer. And this is just an example of how that test helped us with this. So again, in that survey that was conducted in 2015 and 16, this is divided by regions of the state. You can see that 100% of the females reproduced on PI88788. It's hard to see, it's that third one. We did see some with well, that's a whole other can of worms, but we did see some that could reproduce on Peking, which is a little concerning because we're not using Peking in the state. So I would say Peking is good, Band-Aid for now, but again, not a silver bullet for this. So in summary, we are losing availability of PI88788. What is the availability of Peking? This is something we really need to stay on top of. You know, when I think about what Mark and some of the other weed scientists are doing with the lack of chemicals, it's going to be harder to get at the nematodes than it is maybe on some of those weeds, and it's already hard to get on the weeds, right? The illustration I like to use is in Missouri, Dr. Bradley's working on weed electrocution, right? 20 years ago, if Dr. Bradley told you he was working on weed electrocution for water hemp, everybody would have laughed at him and been like, what? And now it's not that off. And so just keeping in mind that this is something we want to manage up front, as opposed to have to pay for in the end. Seed protectants may offer protection. They can impact yield in a positive way. New tools cannot be silver bullets. And again, get a baseline understanding of the SCN levels in your field. And so with that, I'd be happy to take any questions. Thanks for your time today. I appreciate it.